We are the spring of 2018 Associated Biodiversity Group. Our group tested the effects of 3 different management systems on associated biodiversity that can be observed on site. The 3 different management systems differed by how they were treated over winter when no crops were grown. These 3 systems were a field left fallow, a field with cover crop, and a field with cover crop that was grazed by sheep. We then defined some variables for associated biodiversity that we wanted to measure. We will now take a quick moment to explain what associated biodiversity means.
Biodiversity in agriculture can be both planned and unplanned. Associated biodiversity refers to the unplanned biodiversity of organisms within a system. This includes other plants and animals, macrofauna and microfauna. It is important to note that we do not regard associated biodiversity as simply weeds or pests.
So instead we use the terms resident vegetation to refer to plants that were not intentionally planted, and macrofauna for the insects and annelids observed. From here we categorize sampled individuals as either beneficial or detrimental given the specific context. Within resident vegetation, we measured the percent canopy cover, diversity of species, and dominant taxa of each treatment. For macrofauna, we measured relative abundance of worms, relative abundance of above-ground arthropods, and the diversity of sampled individuals.
A winter cover crop was planted in November on 2/3 of our experimental field. The north end of the field was cover cropped, consisting of 70% orchard grass, 15% red clover, and 15% ladino clover. In late February, the south part of the southern side of the cover crop field was mowed. At the same time, the northernmost portion of the cover crop field was grazed by 16 ewes for 12 days. The 3 treatment plots were prepared on the 1st of May. Pearl millet was planted 14 days later. The millet has been fertilized and received furrow irrigation. The millet will be harvested this summer.
Hypothesized that the cover crop and cover crop graze treatments would prove to be similar in their encouragement of resident vegetation, resulting in no statistically significant difference in canopy cover or species richness. However, both cover crop and cover crop grazed are predicted to have greater canopy cover and species richness than the fallow field. Furthermore, we predict that cover crop and cover crop grazed will have a similar top taxa profile, that is, the most abundant species, which is most likely the mixed cover crop species that were planted, while fallow will have a different top taxa profile that will consist of resident vegetation species. Weed sampling began on April 18th and continued on May 2nd, 16th, and 23rd. Throughout our sampling, materials used include a 2 feet by 2 feet square frame, a smartphone with a Canopio app and photo-taking ability, weed identification cards to facilitate identifying our weeds, a data sheet, and a pen. To begin, sampling locations were randomly selected in the fallow, cover crop, and cover crop grazed treatment fields by tossing a square pipe frame across the field and sampling from the site it landed on. On each sampling date, we randomly selected a different sampling location rather than monitoring the same areas over the course of the experiment in order to collect a more varied set of data. Using the Canopio app, we measured the percent canopy cover of each sampling area and recorded the numbers in our data sheet. Each photo was labeled with a sample number and the treatment type for future reference. The app unfortunately did not differentiate between type of green coverage, so seedlings had to be removed manually when we measured after the millet emergence. Within each sampling frame, we collected inventory of observed taxa and calculated the sum of the number of taxa per treatment in order to calculate the richness. We then recorded the top 3 most abundant taxa that comprised over 50% of the overall population. Post-sampling, we categorized resident vegetation as beneficial or detrimental relative to its impact on the crop, taking into special consideration the status of most abundant taxa.
On the first 2 sampling days, we saw a statistically significant difference for canopy cover and species richness between the 3 different treatments, wherein both cover cropped and cover cropped grazed had higher canopy than fallow. On day 1, cover crop grazed had a lot higher cover than cover cropped, but this pattern was reversed for day 2. On the last 2 days of sampling, samples across treatments were fairly similar. In regards to top taxa, the cover cropped field had the most diversity, followed by cover crop grazed and then by fallow. In general, it seems that cover cropped and cover cropped grazed treatments have greater overall diversity in comparison to the fallow treatment, which could potentially help with sustainability in a farming system. But since our samples were limited, we cannot say with full confidence that these practices encourage greater resident vegetation diversity throughout the pearl millet growing season.
Our group hypothesized that cover crops provide habitat and food for beneficial above ground and below ground beneficial beings. In turn, pest management will benefit from cover cropping due to an increased population abundance and diversity of beneficial predators and controlled pest populations.
Our soil sampling began on April 18th and continued once the millet was planted and harvested on May 23rd. We gathered 3 samples for the fallow, mowed, and grazed treatment fields. To select each sampling location, we stood in the center of each treatment field and randomly tossed a large soil aggregate into the air and sampled in the place where it first made contact with the soil. Once the location was selected, we used shovels to dig out a 30 centimeter by 30 centimeter by 20 centimeter soil monolith and placed each soil clod into aluminum trays, paper bags, or plastic bins.
Each soil monolith was sorted through by hand, breaking up aggregates as much as possible and using a sieve when necessary. Macrofauna data were recorded and divided among 3 categories: earthworms, Enchiridae, earthworm eggs, and other insects. Once recording was complete, all collected macrofauna and soil was replaced into its original location, and the data was inputted into a shared Google Sheet to digitize and compile the information.
Prior to installing the pitfall traps, sampling locations were mapped out randomly to ensure variability in results. To begin sampling, a hole large enough for the cup to fit was dug and cups were placed underground with the rim above soil level to allow macrofauna to walk across the soil and be captured within. The first cup placed in the soil was weighed down with a rock to prevent it from being moved or lost. A second cup was then placed inside the first cup for capturing macrofauna.
During our scheduled observation days, we removed the upper cup and transported the samples back to the classroom for further observation and recording. Contents were ran through a sieve to separate macrofauna from water that had accumulated due to precipitation. Collected macrofauna were identified using charts and magnifying lenses and divided into categories including flies, aphids, thrips, beetles, spiders, wasps, and another category. All recorded macrofauna was then entered into a digital spreadsheet.
Sampling locations for the sticky traps were placed beside the pitfall in each field for ease and to account for any time constraints. On scheduled observation days, sticky traps were removed from the sticks they were tied to and transported back to the classroom for observation. Using a random number generator, we concluded to use the 10th square of each sticky trap for observation.
Macrofauna captured by the traps were observed using magnifying lenses and were cross-referenced using a macrofauna chart and divided into separate categories including thrips, flies, wasps, leafhoppers, aphids, beetles, spiders, and other category. Following our in-class recording, data was uploaded into a spreadsheet.
Pitfall trap measurements suggest that species richness increased over the course of the experiment for all 3 treatments, with a more significant increase in both of the cover crop fields. In looking at relative abundance of both beneficial and pest species, we found that the occurrences of pests exceeded the beneficial insect abundance in the fallow treatment. Our data also showed that beneficial insect populations exceeded pest populations in the 2 cover cropped fields. In particular, beneficial insect populations greatly outnumbered the pest populations in the grazed treatment. Therefore, we can confirm our original hypothesis that the promotion of associated biodiversity enhances ecosystem services, in this case, pest management. The sticky trap measurements revealed more species richness in the fallow field than the cover crop and cover crop grazed fields. For our relative abundance measurements, we can generally conclude that the planned biodiversity of cover cropping and grazing allowed for better pest management. These 2 treatments seem to have encouraged beneficial above-ground arthropod abundance that controlled pest populations. For our worm abundance experiment, more worms were found in the cover cropped and cover cropped grazed treatments. We found fluctuations in total macrofauna discovered in our soil monoliths, which we can contribute to different soil moisture levels as a result of furrow irrigation. The most worms were found in the cover cropped grazed treatment, followed by the cover cropped, then the fallow. Measurements from later sampling dates yielded higher amounts of worms than the initial measurement. While both cover cropping systems had greater worm populations were higher in the grazed field. The second fallow field measurement did not yield a significant difference in worm population. This supports our hypothesis that cover cropping with grazing promotes greater population abundance.
In recommendation to our future cohorts, we suggest that they develop a more concrete plan with a week-by-week schedule prior to beginning the experiment. We also recommend they utilize a group document and keep it updated throughout the course of the experiment to account for any changes in methodology or dates of plan management procedures. A more general piece of advice for our next cohorts is to be prepared to be adaptable and willing to work with the TAs to ensure the success of the experiment.